Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled From Concept to Clinic, Overcoming the Challenges of Potency Assays in Cell and Gene Therapy Development. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today is Audrey Chang and Joe Newcomb, who will discuss the complexities of potency assays in cell and gene therapies, highlighting regulatory guidance, assay development strategies, and real-world case studies to ensure clinical success and compliance. After the presentation today, we'll have a live Q&A session, so do feel free to drop your questions into our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to these during the session today. I'd also like to draw your attention to the Resources tab on the right-hand side of your screen, where you can find more information on the topics covered today. Now I'd like to introduce our presenters. Audrey Chang, with over 28 years of experience in biological product testing and laboratory management, now supports clients at WuXi Advanced Therapies, leveraging her expertise in biologics and novel therapies to drive innovative products to commercialization. Joe Newcomb, Senior Director at WuXi Advanced Therapies, brings over 20 years of experience in microbiology, quality control, and rapid testing to drive innovative solutions for cell and gene therapy manufacturing and product safety. So without any further ado, I'm going to hand it over to Audrey to kick off today's presentation.
Thank you, Abby. Good morning, good afternoon, everyone. Thank you for joining us today. I am struggling with a head cold, so I apologize in advance for sounding like a frog today. Okay. So WuXi AppTech supports our clients in advancing discoveries and bringing new medicine to market as a global leading supplier of R&D, manufacturing, and testing services. And the 4 divisions pictured here provide integrated end-to-end services for both contract research and contract testing, development, and manufacturing, supporting over 6,000 clients worldwide. Both Joe and I are from WuXi Advanced Therapy, located in the lovely Navy Yard in Philadelphia, Pennsylvania. So pictured here are the 4 buildings that make up the Philadelphia campus. The 3 bottom pictures are our manufacturing facilities, and we support process development, early phase clinical programs, as well as late phase and commercial cell and gene therapy programs. Now, the 4th picture on the top right-hand side of your slide is a building. It's our testing facility in Philadelphia, and that is located just across the street from all of our manufacturing facilities. This building, all 140,000 square feet, was designed and built to do one thing, and that is quality control testing. So in this building, we test advanced therapy products from our own manufacturing sites, as well as provide standalone testing services for both advanced therapies and traditional biologics, such as monoclonal antibodies, vaccines, and recombinants, all in accordance to CGMP requirements. We find that by co-locating testing with manufacturing, we have created an ecosystem of top-notch scientists and quality experts who collaborate each day to deliver results for our clients. So today, what Joe and I would like to share with you is our experience with potency assays as it pertains to cell and gene therapy product. Potency assays represent one of the most challenging and most rewarding areas for us. So I will set the scene with an overview of the points from a recent FDA draft guidance, and Joe will share some real-life experience, also known as pain points and lessons learned for assay development and tech transfer for the road to GMP potency lot release testing. So very quickly, as everyone should know, quality control testing is an integral part of all GMP biologics manufacturing, as it ensures product acceptability by generating data that meets scientifically based specifications, as well as demonstrating overall control in a manufacturing program. QC is mandated worldwide by global regulatory authorities, and although minor differences may be present, they do require analytical insight into the following questions listed on the right-hand side of this slide. Identity, have you produced the correct product? Purity, what manufacturing and product byproducts are present in the final drug product? Potency, the topic today, and quantity, how much have you made and how well does it work? Safety, is your product free of unwanted adventitious agents such as viruses, mycoplasma, bacteria? And finally, product quality attributes. Have you demonstrated that the product specific moieties are present and in the correct amount? Okay, so modern biologics really have maintained a strong safety record, and that is really due to good CGMP manufacturing practices and testing that fall in three distinct categories as shown on the right-hand side. You have to characterize your starting material, including the cell bank or raw materials, anything that goes into the process. You have to perform batch release testing, a panel of assays that is conducted at prescribed testing points, such as the unprocessed bulk, the drug substance, and the finished product. And data from a documented clearance studies is used to test the downstream purification process's ability to remove or inactivate potential contaminants. And data generated from these three categories, sometimes referred to as the biosafety testing triangle, will provide assurance that the biological product, whether it is a monoclonal antibody made in CHO cells or a complex allogenic cell therapy product, is safe. This three biosafety triangle approach should apply to both. However, it is well known that cell and gene therapy is actually much more complex than a mAb made in CHO. And this complexity and diversity of the manufacturing process introduces many new considerations that must be taken into account in a risk assessment. So your starting materials, selecting unique starting materials such as plasmid or leukopak, you're going to require comprehensive testing and screening strategies even at an early phase. You're going to make critical decisions during in-process, and that's going to be made on data generated from in-process tests, which require a quick turnaround time, robust results from a variety of sample matrices. And finally, as with all biologics, the drug substance and drug product needs to undergo QC release testing to ensure the product safety and product characterization. Now, potency assays are a key part of the product characterization. A potency assay needs to provide quantitative data that not only reflects the product quality, but ideally represents the therapeutic mechanism of action. But this mechanism of action, this MOA, can be complex, multifactorial, or may not even be fully understood. So it is important to develop and share a strategy of how you're going to deal with potency with the regulators early in the process. So the FDA, helping us, released a draft guidance late last year, and it's going to update the 2011 guidance that was considered outdated, especially with these new products. And in this document, potency is defined, as you can read here, as a specific ability or capacity of a product to affect a given result as indicated by the appropriate laboratory test or adequately controlled clinical data obtained through its administration in the manner intended. Now, translating that for QC release, what does that mean? For biological products, potency assays are used to measure the biological activity that corresponds to the MOA, as I indicated earlier. And these assays need to be quantitative. It should correspond to the dose. It should reflect the quality and stability of the drug product, and ideally determine the clinical effectiveness. And the possibility of finding a potency assay that ticks all these boxes may not be realistic or even feasible for complex products, even in early stage. So in this draft guidance, this issue is addressed. It actually introduces what I call a paradigm shift, from it's not just one single potency assay that's going to give you everything you need. You might have to have a comprehensive multifaceted, what they call a potency assurance strategy, and it may include a variety of assays across the product lifestyle, including PD, comparability, release testing, and it evolves as much as your manufacturing process does. I'll get into that a little bit. Okay, so I'm going to go very quickly some of the key points in this draft guidance that we here at WuXi Advanced Therapies have looked at. And so some of the key points is that you need to be aware and monitor potency at all stages of the product lifestyle. And as I mentioned, instead of having one assay, you actually have a strategy. It should ultimately reflect the mechanism of action. You should know the mechanism of action, understand this is how it relates to the product CQAs. And as you get more understanding, you should update and refine your potency method. You can use non-clinical studies to inform the initial potency designs and keep that information to move forward. And you may need additional multiple complementary assays and use statistics. It's not always the be-all, end-all, but it's good to look at that as well as a tool in your toolbox. The gap assessment is key. You have not only considered the in-lot release, but understand that there's also items you look post-release, the container closure, delivery device, how you store it, the shipping conditions, and following in preparation. So at the end of the day, it's a product potency impacted by the quality of the process starting material and from healthy donors versus other ones in disease states. When to use potency assays, it's actually not just during lot release. You use potency assays for stability studies. You can consider different acceptance criteria if you can justify it. It's used for in-use studies. It can be used for how a product reacts in different clinical conditions. And then very importantly, process comparability studies, both analytical and process comparability. For validation, nothing really new here. It should follow the ICH parameters: precision, accuracy, specificity, robustness. And the method should be qualified early. You should invest early in your potency assay to eliminate the reject or having it come in late into your development phase. It could be a showstopper. Make sure you use acceptance criteria. Don't like to use report only or for information only, especially in the later phases. Cellular products have an inherent high variability, so you can use that for early, but you should be able to at least collect that information to set specifications later in the phase. And if possible, certainly have reference materials and controls, recognizing there is no compendial method, but taking the time to establish reference material and to performing bridging studies among reference material is probably a good thing to invest in. So I went through that really fast because I wanted to save time for Joe, but just to understand that the areas of a potency assays, the 5 takeaways, right? It should reflect the MOA. It should produce quantitative data corresponding to drug dose. It should reflect drug quality, drug stability, and reflect clinical efficacy. And this is challenging for both industry and regulators. It requires early investment in potency assay development, which will require a balance, between what you want to do and your development timeline. I should also highlight that just last month in November, the FDA released another useful document. They call it the FAQs. It reflects the interactions from their town hall meetings on this guidance. And it goes into more details and gives some practical suggestions that can be useful in your execution. So now I will turn it over to Joe as he can go through some of our specific case studies. Joe?
Thank you very much, Audrey. Good morning, good afternoon, everyone. So as Audrey mentioned, I'm going to go through some case studies that we here at WuXi Advanced Therapies have experienced with potency assays that we've worked with internally, and as well as focusing more on transfers. So as Audrey mentioned previously, that gap assessment is really critical when you're evaluating the method, making sure that you're really looking at the reagents as well as the equipment. Not all reagents are created the same. Some of the areas where we have had some experience was working with international partners where the same reagent isn't available. Excuse me. And really doing that the evaluation of the assessment of the reagents to ensure that they are the same or can be used equivalently within the methods. The same with the equipment. Looking at the version of software can have an impact on data being generated. So the types of things that you want to watch out for. Also looking into documentation. Make sure that the procedure that is being transferred is clear and concise from the transferring site. Where we've run into issues is there's a misalignment on cell counting methods. The way that equipment is being handled or treated is slightly different between the two different sites, as well as data analysis. What type of data analysis is being done? Is it being done on an Excel spreadsheet within the equipment itself? These are the types of conversations and evaluations that should be done during those gap assessments. Some other watchouts that need to be taken into account are cell banks. So if at all possible, the two sites, the transferring site and the receiving site, should really be sharing the same cell bank. Creation of new cell banks just adds a new layer of complications, but it happens. So make sure that any new cell banks are thoroughly characterized before they're used in the methods, with clear recovery and growth instructions to make sure that we're introducing as little variability as possible. And then FBS. Understand the differences of FBS, the grades. Is it heat inactivated versus non-heat inactivated? How is it treated before being used? What impact could that have on the cell growth itself or in the assay as well? And then training. Everyone knows training is very important. So making sure that the receiving lab receives the same amount of training that someone from the transferring lab would get. We recommend having the training lab transfer into our analytical development team, and then we would work directly with our QC team for that internal transfer. So one real world example with some data. So this is just an example of a cell-based toxicity assay with a luciferase endpoint. This assay uses two different target cell lines. One is a wild type, one is a knockout. Over the course of the assay transfer, we started to see some variation in the data with the target cells only. So no product being introduced into the assay for these. It was just the cells growing by themselves. This was a new cell bank, and it did exhibit different growth characteristics from the ones that were used to develop the method. So going back through when we started to see these issues with some assay failures, we had to institute some stricter cell passaging requirements for the new cell bank than what was in place for the original cell bank. As well as we had to take a look at the media and the expiry of the media. We were realizing that the older the media was getting, the less that the cells were responding. So we had to institute a stricter earlier expiry in our hands than what was seen previously. So on the right-hand side is some actual data. The lower bars is the prior to optimization, and the right-hand side is following optimization. So this was the difference between some assays failing the validity criteria versus passing more with a more stringent parameter. So then some of the common assay transfer pitfalls that we tend to see are really the unrealistic timelines. So making sure that both teams align on the priorities, and if speed is a necessity, what impact that's going to have on the method. And then vague agreements. Training is one that I've already mentioned. So making sure that the receiving lab really does get the adequate training from the transferring lab, and it's not just a handoff of documentation. And then material readiness. Is there material, test material that has a known result, a reference control, some type of material where there's an expected result that both labs can compare back to see, is there a difference being seen in the receiving lab versus the transferring lab? And then really the communication. So having a clear cadence of communication between the two labs to more quickly resolve any issues as they arise, instead of waiting until all of the data is available and say, this didn't really work out well. And then as a contract testing and development manufacturing organization, what we like to work with our teams on and our partners on is really striving to not duplicate the expertise and skills, but it's more complementary. Make sure that those agreements, as I mentioned before, are very clear with clear success metrics. What are we looking for? What does constitute a passing run versus a failing run? Making sure everyone understands what the goals are, having that communication, and then having clear roles and responsibilities, particularly who's the point of contact for escalations if there are issues, who is the technical person versus the project person. Those types of things should really be laid out prior to the initiation of a program. And then one final example that I'll point out here. This is a long-term partnership that we have had with one of our partners. And I'll focus here really on the potency assay. So we worked with this client for a number of years to work through, transfer, optimize 3 different versions of a potency method throughout the course of their journey, which ultimately led to commercialization of the product that we continue to support to this day with those with the potency assay that we have worked hand in hand with them. So it is these potency assays are a long-term timeline item. So what you start with is not necessarily what you're going to end with, and it's just an example of something that we have gone through here with some of our partners. And then just to wrap up, so WuXi Advanced Therapies, just kind of walking through some of our capabilities as I've been talking about the potency side of things. We also continue to support molecular virology, micro, and other analytical testing, biosafety testing for global submissions. We are also a market leader in viral clearance. So we perform viral clearance testing quite frequently on site, as well as release testing in cell banking. The release testing, as Audrey previously mentioned, on cell and gene therapy products, but as well as biologics and other products. And then finally, just a range of WuXi's capabilities across the board, from early research and discovery through those the commercial testing across all of our sites throughout the world, focusing on, again, viral vector cell therapies, as well as the testing that we perform for our external partners for the other types of products. And then finally, just to summarize, kind of our bottom line is that we have a robust track record that we demonstrate our ability to deliver and scale our programs, including the testing, and then that integrated testing with our manufacturing partners that are also on site helps to reduce any risks of shipping for testing, as well as helps to accelerate program timelines. And that is wrapping things up, and I will hand it back over to Abby.
Thank you very much to both of you, Audrey and Joe, for that fantastic presentation. We're now going to start our Q&A session, so I invite you both to join me on camera. And just a note to our audience to keep sending in your questions. We'll get to as many as we can in the rest of the session today, and then answer any others via email. So first question here, and this one's for you, Joe. What would an in-use study typically look like?
Sure. So an in-use stability study really needs to evaluate what's going to happen to the product at its final endpoint. So most likely in the clinic, what is going to happen to it? How is it being thawed? How is it being stored? Those are the types of things that need to be taken into account, and what happens to the product once it leaves the manufacturer's hands, and what impact does it have on the activity of that product. So brief kind of overview of what an in-use stability study would look like.
Fantastic. Thanks for that, Joe. Now one for you, Audrey.
Okay.
Could you remind us of the name of the FDA document about the potency assay?
It's just a guidance for industry. It's a draft guidance. It was issued, you just look up FDA. I don't have the exact title, but it's a FDA guidance for potency assays for cell and gene. They might use ATMP or cell and gene therapy products. It was released in December of 2023. And then the one for the FAQ was just released last month.
Fantastic. Thanks for that, Audrey. And staying with you here, are there generic potency methods that could be used for multiple products?
Well, potency by definition is a product specific characterization assay. So I think it's a stretch to have a generic potency assay. But I think what you can do is to shift from assay to more strategy, right? That can be a generic approach. You might start early phase with some of the simpler transcription or translational assays to define potency, and you gain information and then move forward as you do perhaps for the later phase, some of the binding or the more sophisticated ones that Joe did. So that can be a generic strategy, but by definition, it is a product specific endeavor.
Absolutely, that makes sense. Thanks, Audrey. One for you here, Joe. How do you prepare for potential differences in healthy donors versus the patient product?
So that is always a difficult one. Obviously, you're not going to want to use patient material as you're working through doing development of a potency method. Initially, it's evaluating those parameters, trying to take a look of going back to that gap assessment. What could the differences be in those types of materials? And for the types of parameters that you're looking into, try to incorporate that into your acceptance criteria. And then there's always going to be some surprises. So kind of be prepared for what surprises may come if the material from a patient starts to behave differently.
Absolutely. Thanks for that, Joe. And one for you here, Audrey. What's the most common issue that's identified when going through an assay transfer?
I think Joe gave a good overview on some of the surprises that we said. It goes without saying that tech transfer, it's never boring. It's never without surprises. So to have that understanding that it's not going to be a straightforward, check the box type of exercise, and as Joe emphasized, you have to really not underestimate the importance of clear and effective communication, and having that cadence of meeting, that open communication between our excellent analytical development scientists with your scientists really just represents best practices, ensures the probability of success.
Thanks for that, Audrey. And in response to an earlier question, someone from the audience has chimed in that the FDA guidance is called FDA Guidance Potency Assurance for Cellular and Gene Therapy Products. So thank you for that.
Thank you, whoever saved me on that one.
You don't have that memorized, Audrey?
One for you now, Joe.
Thank you to our audience for that.
Joe, how would you transition from one lot of reference material to a new lot?
Yep. So Audrey did mention it as she was talking, but trying to make sure that there is some material from the old lots available to do that side-by-side comparability testing, a bridging study to make sure that they behave the same way. If it's just different aliquots of the same lot, are the results expected to be the same? Or if it is a different lot, making sure that, yep, it's going to behave the same way, but also evaluating it for, is the range going to be slightly different? So some of the things that we keep in mind here is making sure that we're building in the idea of that bridging study between the different lots.
Absolutely. Thanks for that. Lots of questions coming in, so I think I'll squeeze in just one more here. But feel free to keep sending in your questions and we can take anything else offline. Is relative potency assay recommended for cell and gene therapy products?
Audrey, do you want to start? You want me to take that one?
You take that one.
Okay.
My answer is it depends, but you go ahead.
Well, yes, the same. And then again, relative potency really does come down to having a well-characterized reference standard or control to compare back to. So in the absence of that, it's really difficult to have a relative potency method. So those early phase programs, typically they're not relative potency. Some later phase do once they've gotten enough material to characterize a reference standard that routinely works the way it's expected. So that can be a little bit more difficult and time consuming across the program.
So I would just emphasize also, and I know it's hard because you're making small scale. Every drop of your material is precious, right? But you have to keep in mind that keeping some of these retains early on, it should be part of this strategy, right? That you keep that so that you're able to bridge as you move. You refine your manufacturing process with data each time. That type of mindset should also be applied for potency assays. As you gain more knowledge, you refine and you come up as you move forward. So that would be the other thing I would say.
Absolutely. Thank you both for that. And thank you for all those answers. That's unfortunately all we've got time for today. But any questions that you didn't get to, we can take offline and reply via email. The webinar will be available on demand tomorrow, so do look out for an email from us with the link. And we'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources tab on the right-hand side of your screen or in the on-demand email. And all that's left is to thank Joe and Audrey once more for a great presentation today. And thank you to the audience for listening. We hope you'll join us again soon.
Bye.
Thank you.